Our second talk of the day is going to be about acoustic seascapes, I think. Over to you. So we've got Lloyd Izard talking to us. So thank you, Lloyd, and over to you.
Thank you, Adrian. Thank you, Elisa. Hi, everybody. My name is Lloyd Izard. Just one second. I show you here. My name is Lloyd Izard. I work at the L'Océan Laboratory in Paris, in Sorbonne University. And today, indeed, I'll be talking about acoustic seascape. And more precisely, I'm trying to understand how we can have a better representative view of the pelagic fauna variability by integrating multiple frequencies and circadian variability within pelagic fauna biogeography. So this talk will cover one paper that's actually under review, and also I'll present some ongoing work regarding circadian variability. And before I start, I'd like to thank my co-authors for their great contribution to these complementary topics. So today, for this presentation, our area of interest will englobe the epipelagic and the mesopelagic zones. And as you know, these layers are home to a wide array of pelagic fauna, such as crustaceans, fishes, and diverse gelatinous life forms. And as mid-trophic levels, they really allow the transfer of energy and biomass along the trophic web. And we know as well that there is a large biomass in these depths that can support, in turn, top marine predators. And we have, for example, the largest fish stock in the world. But how important is still quite uncertain. As you can see here, we have different orders of magnitude in biomass estimate, depending on the method that we use. And any of these estimates actually represent important biomass, but for now, these different numbers can get us lost in translation. And a second major characteristic I wanted to cover today is the deal vertical migration of these animals. I'm sure you're all familiar with this pattern. And I wanted to highlight that really this migration connects vertically and daily the different layers of the ocean. And that has many implications. For instance, this daily biomass redistribution has implications on trophic interactions. And we know that, of course, there's also impact on biogeochemical cycles, and that can be part of the active flux of the biological carbon pump. So we really need to see these deep ecosystems as ecosystems in perpetual motion, as you can see here on this image from NASA. But this perpetual motion of the system actually complexifies the identification of spatial patterns, and even at a global scale. So now, how can we monitor these ecosystems? Well, traditionally or historically, one way of doing so is to use nets to sample these animals, and that will give you direct information on species identification, or as well on their distribution, and as well their biomass and even their morphology. And with this information, you can actually identify ecoregions, as this map proposed by Sutton and their collaborators in 2017. And these ecoregions are indicators of spatial scales at which faunal communities are expected to be broadly similar in composition. Now we can mention that there remains a major gap in net sampling, and that's due to the difficulty of working in deep sea, and that can limit our understanding of the fauna distribution at a global scale. And we can also mention the inefficiency of trawls in representing the actual proportion of species below the ocean surface. And that's one limit among other I can name here. And as complementary tools, many new devices have emerged over the last decades to try to monitor these dynamics. And today we'll focus on the active acoustics. So it's a method that emits a sound wave in the water column, and it will then measures the echo it gets when it encounters a target. And you can represent the data as an image here, or as well as a curve, and the peak of the curve here indicate the depths of the animals in the water column. You can then represent an image, and we call that an echogram that you can see here. And that's one of the first echograms to have been published in 1948 by Johnson. And in his paper, you can also read that direct census of population are time-honored methods in ecological studies and will always be part of such investigations. However, any physical means that provide a shortcut to useful generalizations, such as relative concentrations and activities in space and time, serves as real need. And that's exactly what active acoustics is about. And it's also one of the only tools that allow to monitor mid-trophic levels dynamics with such a spatial, vertical, and temporal resolution. So nowadays, the data that we have, the echograms look more like this. And on this image, you can identify sand scattering layers of animals. And also you can observe vertical migration patterns that I mentioned in the introduction. And as you can see on this image, the main limit of active acoustics is the low taxonomic resolution it provides. And one way to overcome this limit is to use multiple frequencies that will depict complementary acoustic seascape or different vision of the water column. And that's due to the frequency response of the animals. You get here on the y-axis the echo or the target strength as a function of the different frequencies that we commonly use in hydroacoustics. And for instance, the type and size of fish swim bladders will greatly impact their detection by acoustics. So that's something to have in mind. And over the last decades, again, there's been a growing number of acoustic observation, and that have allowed us to describe acoustic seascape in major sectors of the global ocean, as you can see here on this image from Clefger and the collaborators. And from such signal, and at global scale, it was recently introduced the notion of echo biomes, which are acoustic seascape representing sand scattering communities with comparable structural and functional properties. From the signal also, from these regions, you can compute a proxy for integrated biomass distribution. And you can see here that you have regions with large biomass at the equator in subtropical regions, and other regions such as the oligotrophic gyres with low biomass. Now, I think we can identify two main limits with current acoustic biogeography. And the first one is that until now, the research have principally focused on the 38 kilohertz frequency to perform biogeography or biomass estimates at a large scale. And you can see here on this map that, for instance, we observe and we predict as well the lowest biomass in the Southern Ocean, for instance, or in polar regions. And we can wonder, how is it that we have the lowest biomass in regions that host among the largest population of marine top predators in the world? So that's something we will try to cover during the first part. And the second part is we will try to see how we can integrate temporal variability in pelagic biogeography. And actually, if you read this paper from Sutton and the collaborators, you can read that work remains to be done to produce a comprehensive and robust mesopelagic biogeography, meaning reflecting temporal variation. And that's one limit that I mentioned as well in the introduction previously. So that's it for the introduction. I propose we jump in the first part of this talk, and we'll focus on this low acoustic biomass observed in the Southern Ocean I just mentioned. And actually, we know that this acoustic signal does not match with net-based pelagic biomass estimates that we have observed in this region. And one documented explanation actually is that there is a change of communities, and particularly of fish communities, that impact the detection of animals, and is not an actual loss of biomass towards the Southern Ocean. So we can wonder if our understanding or current understanding of mesopelagic biomass distribution is affected by the choice of acoustic frequency we can use. So to start with, I'd just like to mention that several studies have documented this decrease of acoustic density in different sectors of the Southern Ocean. For instance, Escobar Flores and the collaborators has observed this distribution of signal in the Southern Pacific Ocean. Dornan and the collaborators have observed the same process in the Southern Atlantic Ocean, and that's over recurrent cruises, so over many years. And today I'll provide some information or description of the acoustic landscape in the Indian sector of the Southern Ocean. So as you can see here with the sea surface temperature map, it's really a region that is contrasted with the Southern Indian Ocean to the north and the Southern Ocean to the south. And this transition is marked by the presence of intense fronts, such as the subantarctic front here, or the polar front as well. And we know that the acoustic signal at 38 kilohertz in this region respond as well to these change of temperature, with the lowest values of acoustic signal found south of the subantarctic front. And you can see that on this echogram from Bergel and the collaborators that show the mid-trophic level distribution between Reunion Island and Kerguelen archipelago. So here for our study, we'll benefit from a data set that allows us to have a regional approach was both 38 kilohertz, but as well 18 kilohertz frequencies. And the data was collected during 3 consecutive cruises from the Themisto program that took place in Austral summer between the years 2021 and 2023. And here I'll present some results on the daytime seascape. And each of our observation here, of our dot on this map, is actually composed of 2 profiles. And again, the shape of the profiles indicate the depths of animals in the water column. And as our data set is composed of curve, we can apply functional data analysis. And more precisely, in the study, we applied functional principal component analysis. And I will try to describe it really simply here, but it's a method that allows you to describe each of your observations relatively to a mean observation. And here the mean observation is 2 curves, 1 for each of the frequencies. And we will just place this mean observation on a factorial map, and it will serve as reference for the other observations. So we can project these observations by computing their principal components. And here each of these points is 1 observation, which means it's a pair of curves at 18 and 38 kilohertz simultaneously. The position on the map of this point indicate actually vertical deformation of the pelagic system with respect to the mean observation. And if your point is in the positive part of the x-axis here, the deformation of the observations will tend to have this red-shaped curve, and you'll get the opposite information in the negative part. You can then project the values of the principal component on the map, and you can actually observe a latitudinal gradient for this mode. And you can, of course, do the same for subsequent mode here on the y-axis, and that will give you complementary information on the vertical structuration in this area. What's done generally after this first step is to cluster these principal components, and you can get groups. And these groups here are purely acoustic groups that are based on the vertical distribution and intensity of the signal at both frequencies. And as you can see, we observe here 4 groups in our region. And if we project these groups on a map, we can see that they are spatially coherent with 2 groups north of the subantarctic front and 2 groups south of that front. What's interesting now is to look at the result of the clustering. And to do that, we can compute a mean vertical distribution in each ecobiome, if you want. And here we observe, indeed, different vertical distribution between the groups at 38 kilohertz. And we can compute the same for the 18 kilohertz frequency. And actually, both frequencies might seem quite familiar at first, but we can compute the difference between the 2. And that's what you get in this subfigure here. And what is important to note is that there is a dominance of 38 kilohertz in the 2 northern regions. That's going to be the negative values here on these 2 subfigures. And in the southern region, you'll get a dominance of 18 kilohertz along depths. So that really shows that we have indeed differences in the vertical structuration between frequencies in our system. And we wanted to further understand how these patterns could be shaped by oceanographic conditions. So to do that, we applied random forest methods and acoustic profiles reconstruction. And I will not go into details here, but I'm happy to discuss later on if you're interested. And that has allowed us to compute a proxy for biomass again. And indeed, we observe low biomass at 38 kilohertz into the Southern Ocean. So that's consistent with recent research at the global scale. And what we show at 18 kilohertz is that we reveal a totally new acoustic landscape, and we observe indeed large differences in the distribution of pelagic biomass as a result of sonar frequency choice. And actually, we can dig deeper into these results, and we can compute the difference between these 2 maps, and we observe a shift in acoustic dominance between the north and the south of the subantarctic front. And this shift in acoustic dominance actually matches with change of resonance that was observed by Chapman and the collaborators 50 years ago. And our results highlight that the loss of signal at 38 kilohertz throughout the Southern Ocean is mainly due to a change of community and is not due to a loss of biomass. So to conclude this first part, I think we can say that the frequency choice influences our view of pelagic fauna acoustic biomass distribution at a regional scale. And more precisely, there can be large differences for a given region between 2 frequencies. Here, for instance, we have 2 times more acoustic biomass at 38 kilohertz in the northern region compared to the 18 kilohertz. But the differences can be even more important across the subantarctic front at 38 kilohertz, where you observe more or less 4 times more backscatter to the north, while the difference between the 18 kilohertz backscatter in both regions is quite marginal. And we don't know yet what impacts these differences could have on biomass estimates at a large scale, but that's something we could and should investigate further, I think. Something else we can mention is that there's been recently an area of ecological significance that was identified within our study area around the subantarctic islands here of Kerguelen, Prince Edward, and Crozet. And if we report this region on a map, we observe actually that it's a zone dominated by the 18 kilohertz frequency, and that emphasizes further the need for multiple frequencies to understand trophic relationships in this region. And finally, from a qualitative perspective, the frequency response that we observe could be quite valuable to track different biogeographic regions and transition. Let's get into detail. And that brings me to my second and final part, of course, and that's how we can integrate circadian variability within pelagic fauna biogeography. So from the previous study, I mentioned that we work with a daytime dataset. And here's the result for the nighttime dataset of the clustering. And you can see that the distribution of the groups are quite similar, actually. But if you look into the different curves that we can compute, there's actually a redistribution of the biomass that takes place. That's due to the geovertical migrations. And that leads to an even more complex change of dominance along the water column. And at the regional scale, this leads to different ratios of dominance as well. And that can slightly change the spatial distribution of biomass that we mentioned for the daytime seascape. So these temporal differences is why I think we need to define biogeographic zone with at least both daytime and nighttime vertical structure. And if we go back to this paper from Harris and the collaborators, that's what they propose. And that's what innovative here, I think, is that they define one pixel or one cell of this map as the joint daytime and nighttime vertical distribution of pelagic fauna. And something that got me wondering is that still for these ecobiomes, we do not integrate migration patterns. And if you read this paper from Ungard and the collaborators from 2013, you can read that in the context of acoustic measurements, geovertical migration may actually assist classification. So if we try to summarize the challenges that acoustic pelagic fauna biogeography is facing, we could ask, how can we identify relevant modes of variability that integrate at the same time multiple frequencies, the vertical distribution of the fauna, and as well the nocturnal cycle. So for this final point, we'll start with an example data set, and it's still in the same area in the Southern Indian Ocean. And we have 3 stations, A, B, and C, that were sampled over time. And you can observe here for station B that you have variability between frequencies, and that's due to the frequency response of the animals, as we mentioned. Then between frequencies, you also have variability, and there's going to be spatial variability. And then within one station, you have variability, of course, and that's due to the nocturnal cycle. So on this data set, I will apply now functional data analysis as we applied previously. And we can do that because, again, these images are composed of data that can be expressed as curve. And the goal here is to compare these curves together and identify the depths that maximize the variability of the system. So we apply functional principal component analysis, and I will not go into details again, but that's the result of the analysis. We get different factorial maps that have to be investigated with the different eigenfunctions or different vertical modes of variability that are associated with these axes. We will just now cluster these principal components, and if we cluster the first factorial map, we can actually identify temporal opposition independently of spatial variability of our different stations. What is interesting to me is that if you cluster the second factorial map, you can identify spatial opposition between the stations, and that independently of temporal variability that you can observe. And that really highlights the complexity of the system, because both classifications could be considered pertinent depending on the research question that you're posing. And our question here is, how can we integrate both these patterns within pelagic biogeography? So to do that, I propose to fix the temporal dimension by representing all principal components of one station over a 24-hour period. So that's what you get here. And that summarizes the variability of station A over 24 hours. So the combination of these 5 principal component series is unique to a station or a region or province, depending on the scale that you work with, and it can be clustered and compared to other locations. And that allows, this representation allows to integrate vertical variability of the system and as well multiple frequencies. That's with the functional PCA. And this representation allows us to integrate here as well the nocturnal cycle. And actually, if you use the linear combination of eigenfunctions and principal components, you can actually reconstruct echograms over 24 hours period. And that's what you get on the right here. And this allows me to mention that the echograms that we get on the left here, they do not exist in nature as they combine successive temporal events that cannot occur simultaneously. It's just a vision that combines space and time over 24 hours at the same time. And if you want to unfold time, that's the type of echogram that you'll get. So we applied these methods to Mediterranean data sets from the BioSword cruise that took place in May 2023. And we actually found that the nocturnal variability could be associated with water mass and fronts. And we have different clusters here of the stations with the points that are colored, and as well the chlorophyll A satellite concentration that is shown behind. And for this blue cluster, for instance, we have one specific nocturnal cycle that is identified. And if you look at different clusters, you would get different dynamics, and that would mean different migration depth, for instance, or different migration speed over the different area. And these differences can occur, as you can see here, at really fine scale in our frontal regions. So now what to take home from this second part, because there's been a lot of information, I think. Well, firstly is that we can cluster nocturnal variability spatially, or we propose one method to do so. But for now, we're working at a regional scale, and we can wonder how can we integrate this nocturnal cycle for large-scale biogeography. That means how can we have this ecobiomes representative of nocturnal cycles for each of the different cells here. So for that, we actually need an international and optimal effort of data collection. So why international? Well, because to produce such ecobiomes and global ecobiomes, we need data sets that covers many regions of the ocean, and that's only reachable through collaborative efforts of the different oceanographic fleets around the world. And why optimal? And I found this point quite interesting. Well, because here is a map that shows in gray the different points that were used to do this map here from the French fleet, and you can see in black the different regions where the data was not usable because the sonars were switched off. And here it shows like the loss of signal that we did not get because the sonars were switched off. And that's a lot of information for the Atlantic mid-trophic level distribution. And for other regions, for instance, you get some season variability or day-night variability that would have been covered with such information as well. So actually, this effort to gather international sonar observation has been recently supported by the ICES Working Group on Fisheries, Acoustic Science and Technology, the FAST, and more precisely, and more recently as well, the ICES Working Group on Global Acoustic Interoperable Network, led by Tim Ryan and Alejandro Ariza. And if you have any past or future sonar data that you want to share, or if you're interested in any of this work, don't hesitate to contact them, or you can also join the GitHub page that you can find here at this link. So to conclude this talk, we can just say that during the first part, we've seen that large differences in the distribution of pelagic biomass can result of sonar frequency choice. And then in the second part, we've looked at how we can integrate circadian variability within pelagic fauna biogeography, but this part is still in progress. And to conclude, I think we can also mention that Jet Zone is a great opportunity to gather experts from different domains, so acousticians, ecologists, biogeochemists, physicists, and many more, to study these deep systems over different spatial and temporal scales. And for us, it's also important to collaborate to understand what impact can our research have on biogeochemical fluxes or cycles. And with that, I think I'm done, and I'll thank you for your attention. Thank you.
Thank you very much, Lloyd. Another fascinating talk, lots of information there, as you said. So once again, if people have any questions for the speaker, please raise your virtual hand or just open your microphone and ask a question. So while people are thinking, I'll get one going. So I suspect this is a difficult one, Lloyd, but anyway, I'm interested in your thoughts. So it was quite a striking difference between the two frequencies, the 18 and the 38 kilohertz, as you move south into the Southern Ocean. Do you have any feeling for what that's telling us about the communities in terms of, what are we seeing? Are we seeing a shift to, away from fish towards squid, for example? Do we have any information in terms of what's happening in the Southern Ocean that is distinct from elsewhere?
Yeah, well, the limit in our paper is that we don't have nets information, and that's really a key point that we don't have. But we really based our reflection on the paper from Donan and the collaborators, and they have shown that it's a change of myctophids community towards the Southern Ocean that has a loss of gas-filled swim bladders, and that's why they are not detected. Right. And so we just as well added to this hypothesis or this explanation as well the role of Gonostomatidae or cycloton fish species, which are really small fishes that are highly resonant at 38 kilohertz. And we know that only a few species of cycloton will venture into the Southern Ocean. So we really focus as well our explanation on these two families and on their morphology and swim bladders and size. And we discuss really briefly the role of gelatinous organisms such as salps or siphonophores and so on, but we have really not so much information on these animals and on their distribution. So it's quite complex to really attribute what we're observing here to some communities. But the major explanation for now is the change of fish community and their internal swim bladders, for instance.
Very interesting. Yeah, and I guess the fish species you're talking about are incredibly abundant ones as well.
Exactly.
Yeah.
And as well, the cycloton species, they've been observed to not migrate so much. And on the 38 kilos, I don't know if you paid attention, but there's these deep scattering layers that do not migrate.
Right.
And these permanent layers could be attributed or part of the signal to cycloton species, whereas most of the myctophids do migrate, or a big fraction of them actually. So that's more attributable to the 18 kilohertz. But again, we have to be precautious here, but just hypothesis that we work with.
Yeah, thank you. Thank you. Anybody got any questions or comments for Lloyd?
Excuse me, I have a small question.
Yes, carry on.
Thank you, and it's a very interesting talk. And do you have some information about the organism size, the difference between the 18 and the 38 frequency?
Yeah, the size of the animals. Yeah, let me think just briefly on this. I think I have if we have 1 second, I have something to show actually, I think, that could answer this.
Yeah, I mean, if you're quick, so we're close to the hour, but yeah, if you're quick, I'm sure we can cover it.
Yeah.
Yep. Okay, it's right here. Sorry for that. I've just added that to my slide right now. Can you see my screen?
Yes.
So you can see why does frequency response change across the subantarctic front, right? This one? Or is that what you can see here with the two curves?
Yeah, I can see a graph of fish standard length against target strength.
Yep. Okay, exactly. Yeah, sorry. So indeed, this explanation was based on the fish tender length, indeed. And so for the 18 kilowatts, we do observe bigger fish in size. It is always a ratio of size and swim bladder size as well. And so the 38 kilowatts really dominates for small fish, as I mentioned, the cyclodon species, whereas the 18 kilowatts could identify more bigger fish actually. But in our paper, we have these figures here and more explanation that maybe could help you understand and answer your question as well, if that makes sense.
Okay, thank you very much. That brings us neatly to the end. So the one other thing I would say, Lloyd, I think your idea of using the Jetson community to try and get people to collect this data where they might be able to is a great one. If you want to send me something that I can put in the newsletter or I can put on the web page to try and get people to pay attention to that, I would be delighted to put that out.
Amazing. Yeah, let Andrew know, and I think they'll be delighted as well to share that. Thank you.
Excellent. So that brings us to the end of another call. Thank you very much to Lloyd and also to Elisa, who spoke earlier. The next call in this time zone will be in September, but hopefully there'll be an advert before long for a call in the Southern Hemisphere or the Pacific time zone, which will take place in August. Otherwise, I hope you have a good summer, and thank you for joining the call. Goodbye.
You too. Thank you very much, Adrian.
Bye.
Bye-bye.